Before getting into the lysogeny or a lytic cycle, we will try to understand the arrangement of genes in a lambda DNA. So we all know that this particular DNA has got 48,502 bases and has 40 genes. The beauty of this lambda DNA is that the genes are all arranged in clusters, in a functional cluster. For example, head and tail genes, the genes which are required for the generation of proteins of head and proteins of tail region, they are all arranged in one cluster that is present on the left side of the lambda DNA. Now, the regulatory genes which are important in channelizing this to lysogeny cycle, they are located in the central part of it, whereas the regulatory genes required for the lytic cycle are all arranged in one cluster on the right side of it. So in the left side we have the head and tail, and in the right side we have the regulatory genes required for the lytic, and in the central, so we have the lysogeny cycle. We will try to see the important genes involved in that. When it comes to the head and tail region, there are 19 genes are involved in that. So we can call it A, W, B, C, D, E. Now here F1, F2, Z, U, V, G, T, H, M, L, K, I and J. So now this particular region I am going to say that from A to J. So we have all the 90 genes are arranged which are important for the generation of head and tail region. Now when it comes to the lysogeny, we have 7 regulatory genes in fact. So when it comes to the lysogeny cycle, these genes are very important. They are the ATT site, INT site, XIS site, EXO, BET, GAM and KIL, kill. Okay, now these are the 7 genes are very important in regulating the lysogeny cycle part of it. Whereas in come to the lytic part of it, we have 6 regulatory genes. One is C1, C2, C3, CRO, N, Q. Probably, now these are the regulatory genes. Then it also requires genes for DNA replication, that they are regulated by O and P. Then for the membrane lysis, we have the S and R. So altogether 6, 8, 10 genes are involved in the lytic cycle in fact. So in case of a lysogeny, these regulatory genes are involved in the integration with the host DNA. Whereas in case of the lytic cycle, now we are going to follow lot of steps. First thing is going to be the DNA replication. After that is the synthesis of head and tail. Then the assembly is important. Now the advantage of this lambda DNA is that usually the vectors of phage DNA is used to make the DNA library. For example, we have the cDNA library, genomic library, because with this particular one, it is easy for us to do the screening of gene by the help of DNA hybridization. Then the next important thing we have to study in this case is going to be the packaging. Another one is going to be the size selection. Size selection is that I told you that this particular lambda DNA is 48,502, and now this gene during the assembly or also in the packaging, so it has to have exact size 48,502, but it can also accommodate little bit extra because of the presence of jumping genes and all other things. Sometimes this particular DNA can also harbor additional DNA. So now once it has got an additional CSA, how long this length can be? So it can be increased to another 5% more. If it is more than 5%, then the length of the DNA is not suitable for packaging. So if there is a size in the higher side, so what happened to the lower side? So even if the DNA is too small, then it is not going to get packaged. The minimum size which is required is 75%. So now that will form around 38 kilo bases. So now we have the 48 kilo bases Now the 38 kilobases is the minimum. If it is less than 38, then again it is not going to get packaged in. So now the size will always provide us, because this will give us an idea that how the vectors are going to be designed, and if you want to have any clones to be identified, that can be easily done through the size selection. Probably you will understand this once we discuss about individually. Alright, so now we have the lytic cycle. Another one is going to be the lysogeny cycle. So now we will first discuss about the lytic cycle. So when it comes to the lytic cycle, we are going to study this lytic cycle under this heading. So first thing is an early transcription. So even in the earliest transcription, we have two different phases. One is immediate early transcription, followed by that delayed early transcription. So now first thing is immediate early transcription. Another one is delayed early transcription, followed by DNA replication, then late transcription. So now under this, so we are going to study about how the lytic cycle is going to happen. So in order to understand that, so first we will also study, I told you that once the lambda DNA is get inserted, so now under the cos site, now this will become a circular one. So in order to understand most of the regulatory elements as well as the promoters which are important in the generation of the gene products for the lytic and gene for the cycle, so now we can try to understand in this diagram. Now this is the circular lambda DNA. So in this circular lambda DNA, I have marked one promoter. This is on the right side. We call this PR, promoter on the right side. Now this is another promoter which is on the left side. This is promoter L. Now the right and left is with respect to the gene C1. C1 is a repressor gene. We will study this repressor gene in detail. So now we have this repressor gene here. On the either side, to the left there is a promoter, there is a right, there is a promoter. We call this as a promoter on right, PR, promoter on left, PL. Now what happens is that to begin with, in case of an early transcription, a DNA dependent RNA polymerase will bind to this promoter and also will bind to this promoter and initiate the initial early, that is the immediate early transcription in this direction, that results in the production of a regulatory protein N. From this right promoter, so now the transcription happens in these directions and results in the synthesis of another regulatory protein, that is the CRO. Suppose if you study the detailed structure of both promoter L and promoter R is a little bit complex, because now here the DNA dependent RNA polymerase can bind. It can initiate transcription in this direction, that helps in the synthesis of a CRO protein, but it can also bind to the same promoter and can initiate transcription in this direction, that results in the synthesis of a C1 protein. C1 is a repressor protein, I told you. I will discuss that in detail. So now if I want to understand this particular promoter, so now I have expanded this promoter. Now this is the PR, and now this has got operating region, OR, operating region 1, operating region 2, operating region 3. Now what happens? So now the DNA dependent RNA polymerase will bind to this operating region and transcribe the synthesis of CRO protein, whereas another DNA dependent RNA polymerase will bind to this operating region 3 and results in the transcription of a C1 protein. Alright, so now CRO as well as N protein are regulatory protein. How they are regulatory protein? So when it comes to the C1, by any chance the C1 level increases. C1 is a repressor protein. Once the level of C1 protein increases, this will bind to the promoter L and blocks the transcription of N and further transcription. So it completely blocks the transcription once it binds to the PL. And this will also bind to the promoter R. So now what happens is that, so now CRO is also a regulatory element. What CRO does is that CRO will bind to the PR region and blocks the synthesis of C1. I think it is little bit confusing. I'm just explaining that. Suppose if C1 level is very high, what happens? It will bind to the PL and blocks all the expression of this side, on the left side. Whereas C1 will bind to the region in which CRO is expressed. It will not bind to the region in which C1 is transcribed. So C1, what happens? In the PR region, I told you that it can transcribe in this direction and also transcribe in this direction. Suppose if C1 will bind here, then it blocks the synthesis of C1 itself. So that is not the right thing. What C1 does is that C1 will bind to the operating region 1, where it is responsible for the synthesis of CRO. So what it does that it will bind to that specific region and blocks the synthesis of CRO. So now CRO is blocked, N is blocked. So now the other delayed transcription, DNA replication, everything is blocked. So because of the elevation of C1, this will never enter into lytic cycle. So the C1 repressor, that's one of the reasons it is called as a C1 repressor gene in fact. So in order to getting into the lytic cycle, now the activity of C1 has to be blocked. Now that is done by the regulatory element CRO. So what CRO does is, the moment the CRO level is very high, now this will bind to the PR promoter region. Again, it will not bind to the CRO region, but it will bind to the PRM responsible for the synthesis of C1 repressor. So this is the CRO binding region. So because of that, now promoter region on the right side is much more complex. So now this has got a regulatory, that is operating region OR1, OR2, OR3. Now this region is responsible for the CRO synthesis. Now if you want to inhibit this, then the C1 repressor binds selectively to this region. Okay. On the other hand, C1 does not bind to this region. So what happens? Then the C1 protein synthesis will be initiated in this direction. Suppose if you want to block the C1 synthesis, now that one is taken care by the gene product CRO. Now CRO cannot bind here, it has to bind in this region. So now this is the function of a C1 and how the promoter L and promoter R defines. Suppose if you want to have the lytic cycle in place, so now we need to produce good amount of N protein and good amount of CRO protein. First we will discuss the significance of CRO. What it does is that CRO protein will bind to the promoter region and blocks the synthesis of C1. That is very, very important. So now in case of the immediate early transcription, the only protein synthesized are going to be the N, another one is going to be the CRO. And further transcription is blocked principally because we do have a region what is called as a TR1 and TR2, that is on the right side, and on the left side we have a TL. To this there is a host protein called as a terminator factor P. Now this host protein terminator factor P will bind to TR1, TR2 and TL. So binding of these host protein P will block the further transcription. So as a result, we only have the immediate early transcription that results in the synthesis of only N and CRO protein. Then it is the further transcription is blocked because of there is a host terminator termination factor. This will bind to the TR1 and TR2. So what is the regulatory aspect of N? What this N protein does is it will unblock the terminator factor P binding to TR1, TR2 and TL. So it will unblock. So once it unblocks, then the delayed early transcription continues. Now that results in the synthesis of other proteins like O, P, Q. Alright. Now this is the regulatory aspects of C1 protein, how important it is. If C1 is elevated and if it is bind to PL and PR, then it blocks the entire lytic cycle. It will enter into the lysogeny cycle. So if you want to have the lytic cycle in place, now the C1 gene product has to be regulated. That regulation is purely done by the CRO protein. And another one, in order to continue for the delayed early transcription, N proteins are very important. N proteins, because usually what happens, the host terminator factor P will bind to the TR1, TR2 and TL. So the N will unblock the whole process. Now that results in the synthesis of OPQ as well as C2 and C3. So now with this, in the early transcription, what are the immediate events which is going to happen? In the lytic cycle, some amount of N protein is synthesized, CRO is synthesized, OP and C2 and C3 proteins are also synthesized, then little bit of Q proteins. So these are all the regulatory proteins. Now that regulatory protein combines both lytic cycle as well as lysogeny cycle. Now the proportion of C2, C3 as well as C1 and all other things, that will decide whether it will enter into lytic cycle or lysogeny cycle. Now this is a little bit complex. I will try to explain slowly. We know the significance of C1 because if C1 is elevated, then it will block both the promoters. Alright. Now the C1 concentration is modulated positively by the C2 protein. That means if C2 is elevated, it regulates positively that C1 concentration is also elevated. So in other words, C2 is a positive regulator of C1. Alright. So now this C2 concentration is again regulated by a host protease enzyme, what is called as an HFL. It is a protease enzyme. Now this HFL stands for high frequency lysogeny. So now what this host protease does is that this specifically acts on C2 protein and cleaves the C2 protein. When the C2 proteins are cleaved, then what happens, it will not have a positive regulation of C1, then the C1 concentration is automatically decreases. That is the effect of this one. So now since it is a host protein, if this host protein is not taken care, then automatically it will lead to lysogeny. If you want to get into the lytic cycle, now this HFL protease has to be inhibited. Now this HFL protease is inhibited by the another regulatory protein is going to be the C3. So now I will repeat, C3 inhibit HFL. Because of the inhibition of the host protease, this will not act on C2. So the level of C2 increases. C2 is a positive regulator of C1 gene. Then C1 gene is going to be elevated. When the C1 gene is elevated, it will bind to PL and it will bind to the PR. Then the entire process is blocked. Then it will lead to the lysogeny. Suppose if you want to get into the lytic cycle, now this regulator protein, that C3, has to be decreased. Okay. Once the C3 level is very decreased, since it is unable to inhibit HFL, now the host HFL protease is very active because this inhibitor is in a very low concentration. So once this protease is very active, it will cleave the C2 protein and it is not available for the positive modulation of the C1. Then the concentration of C1 decreases so that it will initiate all other reactions, in fact, so that it will leads to the lytic cycle. Now this is the regulation of C2, C3, C1 along with HFL protein, in fact. So during the early transcription, once we have the immediate and we have the delayed process is completed, so we are going to have a mixture of N, CRO, C2, C3, O, P and Q. O and P, I told you this is very important for the DNA replication, that is the DNA replication. So the next level is going to be the DNA replication. So now I would like to understand, especially in this particular video, that it looks very complex, but we do have a lot more regulatory elements I think you should know the importance of C1, then you should know the importance of C2, C3, then the host protease HFL, then we have a termination factor P here, and these are the TR1, TR2 region. So now what will bind what and how these unmodify the whole process. Now that is a very complex, and this is very, very important. That is the early transcription, that is immediate early transcription and delayed early transcription are very critical. If you follow this particular portion well, then the next one is going to be the DNA replication. So I will discuss the DNA replication in my next video.